The overall goal of qPCRTag analysis is to improve the throughput of the Synthetic Yeast Genome Project PCRTag genotyping assay by implementing a real-time PCR-based detection system and miniaturized reactions. This is accomplished by first using a bulk liquid dispenser to distribute qPCR master mix into each well of a 1536 multi-well plate. Next, a nanoscale acoustic droplet ejection robot transfers template DNA and primers into the wells of the multi-well plate. Then, the plate is sealed, and the reactions are run through a qPCR thermocycler. Finally, the real-time PCR data is analyzed to determine the presence or absence of synthetic or wild-type PCR tags at each locus. Ultimately, the qPCRTag workflow improves throughput.
The main advantage of this technique over the current PCRTag assay is that it allows us to circumvent the use of gel electrophoresis and manual annotation of those gels.
We first had the idea for this method when we realized how time-consuming and labor-intensive PCR tagging on longer chromosomes was going to be.
After culturing and harvesting approximately 2 million yeast cells, add 200 microliters of yeast lysis buffer and pipette to resuspend the pellet. Add 300 microliters of acid-washed glass beads to approximately 1 millimeter below the level of the cell slurry. Then, in a fume hood, add 200 microliters of phenol chloroform isoamyl alcohol. Invert the tube 3 times to mix and verify the cap is sealed. Then, using a benchtop mixer, shake the tube for 10 minutes at room temperature. After centrifuging, transfer 75 microliters of the aqueous phase to a new microfuge tube containing 1 milliliter of 100% ethanol. Invert the tube 10 times to mix. After spinning again, aspirate the supernatant without dislodging the pellet and use 500 microliters of 70% ethanol to wash the pellet. After removing the supernatant, use 75 microliters of 10 millimolar tris, pH 7.4, to resuspend the pellet and vortex to mix. After diluting the DNA 1 to 10, aliquot 30 microliters of diluted gDNA into the appropriate well of a source plate for nanoscale acoustic liquid dispensing. Prepare 850 microliters of qPCR master mix in a 1.5 milliliter microfuge tube and vortex to mix. Then, centrifuge the solution at 20,800 G and room temperature for 2 minutes to remove any bubbles. Then, using a bulk dispenser, dispense 500 nanoliters per well into a 1536 multi-well plate. Collect the qPCR master mix at the bottom of each well by briefly spinning the plate. Next, using the acoustic liquid transfer system and the details outlined in the text protocol, dispense 5 nanoliters of diluted gDNA and 10 nanoliters of 50 micromolar pre-mixed forward and reverse primers into the desired wells of the multi-well plate. Acoustic droplet technology uses sound to vertically transfer small volumes of liquid from a source plate to an inverted destination plate. Here, template DNA and primers are transferred in multiples of 2.5 nanoliter droplets into the wells of a 1536 multi-well plate in order to set up the qPCRTag reactions. Next, use an optically clear seal and a microplate sealer system to immediately seal the 1536 multi-well plate. Immediately centrifuge the sealed plate at 2000 G at room temperature for 3 minutes to collect all reagents at the bottom of the well. To carry out real-time PCR, program the qPCR instrument according to the guidelines in the text protocol and hit Run. Once complete, verify that all the wells pass the master control, which indicates qPCR master mix was detected in the well. Any well that fails represents a potential false negative data point. To analyze the data, visually inspect it within the qPCR software and/or export the data for processing offline from the instrument. In this experiment, wild-type and synthetic chromosome 3 PCR-tag primer pairs were tested with yeast gDNA extracted from 4 different strains, 2 with wild-type chromosome 3 and 2 that encode a synthetic chromosome 3. As expected, synthetic primers generally amplified synthetic DNA and not wild-type DNA. Similarly, wild-type primers produced amplicons exclusively from wild-type DNA. The observance of a small number of false positives and negatives is expected, and possible sources of these errors are described in the accompanying text protocol.
Once mastered, this technique can be finished in about an hour, start to finish. Thanks for watching and good luck with your experiments.